Systemic lupus erythematosus, or SLE, is an autoimmune disease in which the body's own immune system attacks healthy tissue. Cells of the immune system are involved in a wide variety of tasks, one of which is the clearance of debris generated when a cell within the body goes through cell death or apoptosis. The clearance of the cell debris can be performed by macrophages in a process called phagocytosis. However, if not all debris is cleared effectively, it can activate other components of the immune system. Cellular debris can include antigens from the nucleus. These antigens can cause B cells to produce antibodies that bind to these antigens. The antibody-antigen complex is internalized by plasmacytoid dendritic cells, and the antigen is then delivered to toll-like receptors within the endosome of those cells to stimulate the production of type 1 interferons. This can activate such cells, causing them to release large amounts of type 1 interferons, including alpha, beta, omega, and other isoforms. Type 1 interferon can then further drive the expansion of B cells, which can then produce further antibodies that target nuclear antigens. The release of type 1 interferon can also promote the maturation of dendritic cells. Such cells are able to ingest additional nuclear antigens, which are processed and presented to CD4 and CD8 T cells, causing them to turn into autoreactive cells. Type 1 interferon plays an important role in this process via an interaction with its receptor, which triggers an intracellular signaling cascade in cells including T cells, B cells, and myeloid dendritic cells. Levels of type 1 interferon are known to be elevated in SLE, highlighting type 1 interferon signaling as a potential target for the treatment of this disease. MedImmune and AstraZeneca are developing an antibody that can bind to and prevent the activation of the type 1 interferon receptor. By binding to the type 1 interferon receptor, anifrolumab might reduce type 1 interferon signaling within the immune system of patients with SLE, thereby reducing autoimmunity. Research is ongoing to determine the effects of inhibiting type 1 interferon signaling and how this could treat and reduce autoimmunity observed in patients with SLE.